Bio 1440 students, I'm going to go over the example exam in this video for those of you who can't make it to SI, but one thing I want to warn you, do not watch this video if you have not tried to take that exam already. You really need to take it and fail at answering questions so you can do well on the real exam. Watching this video before you take the practice exam is not going to help you at all. So if you want to do well, try to take it with your book shut. See what you know and what you don't know. That is how you learn. Now, the first question. Stomach acid denatures proteins. This allows the stomach enzyme pepsin to break the bonds between amino acids. What kind of bonds is this enzyme breaking? So when you're denaturing, when you're breaking apart proteins, and you're going to denature them so they open up, and you're going to break the bonds between amino acids. Essentially, you're breaking covalent bonds at that point. What kind of bonds are you breaking? If you are breaking those bonds between amino acids using an enzyme, that's a covalent bond that you're breaking. And so more specifically, that would be a peptide bond, which you're expected to know the name of, right? Number 2, how would the properties of water be affected if hydrogen were made more electronegative? So if hydrogen were more electronegative, these are questions where you have to think. This is not something you can just memorize from your notes. These are questions where you have to apply problem-solving skills. If hydrogen were more electronegative, what would that mean? That would mean it likes electrons more. It has a higher affinity for electrons. So that means it's more like oxygen, which means it would share electrons more equally with oxygen. Therefore, less heat would be required to get water to evaporate. So B would be the answer, because water would no longer be polar. It wouldn't form hydrogen bonds anymore. So the next few are the following statements falsifiable. A, yes, B, no. Number 3, all fish in Lake Conway are orange. Yes, that's falsifiable, because if you found a fish that was not orange, you catch a red fish or a blue fish, then yes, it has been falsified. There is no alien life in the universe. Yes, that's falsifiable. You just have to find one example of life outside of Earth, and it would be falsified. There are fish in Lake Conway. That is not a good falsifiable statement. There are fish in Lake Conway. So what would it take to falsify that? That would be finding no fish, right? But if you looked in Lake Conway and you did not find any fish, that's just absence of evidence. So that's not a good way to word that statement, right? Ivory-billed woodpeckers are not extinct. That's hard. That's not a good falsifiable statement either, because to falsify that, to prove that wrong, you would have to find no ivory-billed woodpeckers. And once again, absence of evidence is not sufficient. The moon is made entirely of cheese. Yes, that's falsifiable. Why? Because if you go to the moon and you find a rock instead of a piece of cheese, you have falsified that statement. Number 8. In a single water molecule, hydrogen is attached to oxygen by a covalent bond. How do we answer that? The answer to that is false. In a single water molecule, hydrogen is attached to a oxygen via a polar. I'm sorry. Hold on. I screwed that up. Let me reread. I screwed up my own test. Let me read that again. In a single water molecule, hydrogen is attached to oxygen by a covalent bond. Yes, that is true. Sorry, that is true. Between water molecules is a hydrogen bond. Okay, so in a single water molecule, there is a polar covalent bond. Okay, number 9. Phosphorus has an atomic mass of 31 and an atomic number of 15. How many electrons does it contain in its valence shell? The answer to that is 5. How do you know? Well, the first shell holds 2, right? So it has an atomic number of 15, so you know it has 15 electrons. 2 in the outer shell, 8 in the shell outside of that, and that leaves you with 5 remaining to fill that outside shell. Based on that question, then, number 10, phosphorus will tend to react to other substances. How would you know? Well, you know that it doesn't have a full outer shell, so the answer to that is true. If I could draw on here, I'd draw phosphorus to help you see that, but I can't draw on this PDF. Okay. Number 11. Would the following amino acids be on the outside or the inside of a folded protein? So if proteins fold up, they have a complex structure. Things that like water fold to the outside. Things that don't like water, things that don't like water fold to the inside. Here we have CHC. So first of all, this is the backbone. We ignore the backbone. We only look at the side chains, the R groups. Here we have an OH. O and H, share electrons unequally. Therefore, there's a charge. Therefore, it likes water. So it would be A on the outside. This one, number 12, would also be on the outside. Even though there's lots of carbons and hydrogens here, there's an NH3 with a plus sign on it. That plus sign indicates that it has a charge. It would interact with water. It would be on the outside. Number 13, lots of carbons and hydrogens and nothing else. Carbons and hydrogens share electrons equally. They don't have a charge. They would be on the inside of a folded protein. They'd get away from water. Number 14, which of the following is not correct? I'll let you look at that, but the answer there is C. Single-celled organisms contain organs. Organs are made up of tissues, which are made up of cells. So a single-celled organism could not contain organs. Number 15, when two atoms are equally electronegative, they may interact to form what? The answer to that is D. Nonpolar covalent bonds. They have the same electronegativity. They share electrons the same way. They share them equally. Number 16, hydration shells form when what is dissolved in water? The answer to that is C. Ions. Number 17, if the pH of a solution is changed from 5 to 3, what would be true? There's several answers there, but the correct one is C. Hydrogen ion concentration the concentration of H plus or hydrogen ions is 100 times what it was at 5, because from 5 to 4 is 10 times, from 4 to 3 is another 10 times, and 10 times 10 in this logarithmic scale is 100 times the change in hydrogen ion concentration. And it's increasing because you're going from a higher number to a lower number. That's an increase in the hydrogen ion concentration. Number 18, in sodium chloride, C, water is the solvent, and sodium chloride is the solute. Solute is what is dissolved in the solvent. Number 19, which of these is not a property of life? B, movement. We may not have hit that one too hard this semester. This is an old test, but all life reproduces, has order, reproduction, etc. But not all life moves, so that is the answer. Number 20, which of the following is not a function of proteins? You had a whole list of things that proteins do, but one of these is not on that list, and that one is D, genetic code. Number 21, this molecule is what? That molecule is not a lipid, it's not an amino acid, it's not a protein, that is a carbohydrate. In fact, it is a monosaccharide, right? That's glucose, it's a monosaccharide. And you could look at it here, and this OH is down, you could say, oh, that's the alpha form of this molecule. So if you put a bunch of them together, they would make energy storage. So here, a polymer of this molecule could be used for energy storage. Or if you want to do, if you wanted to include starch and starch and cellulose as an example, we could say energy storage and structure. So two of the above. Though technically speaking, the one shown here is alpha glucose, so only energy storage would be correct. 23. Dogs can cool themselves by panting because of what? D. Water has a high heat of vaporization. When water absorbs a lot of heat, it finally vaporizes, carrying off that heat, thereby cooling the dog. Number 24. Two separate polypeptide chains join together to form what? Well, a polypeptide chain is a chain of amino acids. It folds up into secondary structure, that folds up into tertiary structure, and then when you put two of them together, or two or more of them together, you get D, quaternary structure. 25. The central dogma of molecular biology is summarized by what? What is that core idea of molecular biology? B, DNA codes for RNA, which codes for proteins, which is going to come up again and again in this semester. Number 26, neighboring water molecules in ice can form hydrogen bonds? Yes, that is why ice is less dense than water, because hydrogen bonds lock the molecules in place at a distance from one another, making ice less dense than liquid water. Number 27, which is the lowest level of the hierarchy of biology that can perform all the functions of life and thus has all the properties of life? Where does life begin? That's C, at the cellular level. Organelles are not considered to be alive. They are parts of a living thing. Number 28. What type of bond must be broken in order for water to vaporize or evaporate? Well, when water evaporates, the molecules don't come apart. The Mickey Mouse head stays intact. So hydrogen bonds are what are breaking. Hydrogen bonds. 29. Dietary fiber is what kind of macromolecule? And that's A, carbohydrate. Number 30. Soap binds to lipids and allows them to interact with water. Therefore, soap must be what? Soap must be C, both polar and nonpolar. It interacts with both. It's amphipathic, as you read in your lab manual when we talked about ethanol. Number 31. A gram of which of the following substances would contain the most stored energy? The answer to that is C, lipids. Lipids contain the most stored energy per gram, which is why we use it as energy storage in our bodies, right? Next question, 32. The arrow points at what? What is this right here? That is a C. I'm sorry, that is an A carboxyl group. The amino group, C, is down here. Okay? And here's the side chain, or the R group. Number 33. The amino acid above would be found on the what? The inside or outside of a tertiary structure? Well, let's look at it. There's CH2, that's nonpolar, but there's an OH, that makes it polar. So it would be found on the outside, B. Number 34. Cellulose and starch are both composed of glucose monomers. True. Those are true. One of them is composed of alpha glucose, one is composed of beta glucose. Number 35. As pH increases, the concentration of hydronium, or I didn't use that word this semester, hydrogen ions is how I referred to it. The concentration of hydrogen ions in a water sample will what? As pH increases. As the numbers of pH go up, that means hydrogen ions are going down, so the answer is A, decrease. Number 36. Which of the following is not a polymer? Glucose, starch, cellulose, or DNA? The answer? A, glucose. Another molecule we talked about, so that's a monosaccharide. It's a monomer of a larger polymer. Another molecule we talked about that is not a polymer are the lipids. Fats. They don't have any kind of real polymer. They're just a lot of carbons and hydrogens stuck together for the most part. Number 37. Humans can digest starch but not cellulose. Why? Why? The correct answer is B. Humans have the enzymes that can hydrolyze or break the alpha linkages of starch, but not the beta linkages of cellulose. So understanding that there's a difference in the structure there between those two glucoses, which means there's a change in the properties of their polymers. In this case, cellulose, means we can't digest it. Number 38. Diffusion will cause a cell's internal environment to be more what? Well, let's see. Okay. So that one is a true. We may not get to that one too much on our first exam, but it did show up on this first exam. Okay, 39. Bacteria have a cell membrane. Yes, that is true. They don't have internal organelles like we do, but they have a cell membrane. Number 40. Diffusion moves items along their concentration gradient. That's true. That'll probably be on our exam too. Number 41. What I mean is it'll probably be on exam number 2. 41. The nuclear membrane is fused directly to what? Where's that found? That's found fused to the A, endoplasmic reticulum. Number 42. If a protein were produced and then shipped from the cell, what would the journey most likely look like? Well, it would be made in the rough ER, then go to the Golgi, then go to transport vesicles where it would be shipped out of the cell. Number 43. If an animal is placed in a very hypertonic environment, what will it do? Well, it's going to C, shrink. Once again, that'll be on our second exam. Number 44 also has to do with some questions that'll be on, well, actually, that one could be on this exam, but let me just explain it to you real quick. Number 44, reindeer live in cold climates. If you were comparing the cells found in a reindeer's heart to the cells found in a reindeer's hoof, what would you expect to find? You would expect to find B, the cells to have more saturated fatty acids in the plasma membrane than the cells in the hoof. Why? Saturated fatty acids, as you know, are straight. They compact together really tightly, so you wouldn't want them in your hoof because that would mean your hoof would freeze more easily, whereas if there's curves in them, they won't pack together so tightly. but that's more likely a question for our second exam this semester. Same with number 45, which is false. Okay. Then there are a series of questions. What is this molecule? Number 1. Remember, our test won't have a short answer like this because it's online, but what is this? This is an unsaturated fatty acid. It has a kink in the tail. It's not holding all the hydrogens it could, so it's unsaturated. Therefore, it would be liquid at room temperature. Number 2, what is the hydrogen ion concentration and the hydroxide concentration of water pH of 6? Hydrogen ion concentration would be 1 times 10 to the negative 6, and hydroxide concentration would be 1 times 10 to the negative 8. Why are lipids hydrophobic? Because they contain mostly hydrogen and carbon stuck together, and those have about the same electronegativity. Therefore, they share electrons equally in a nonpolar covalent bond, making the molecule overall nonpolar. Number 4, name the molecule that these would be a part of. This one right here, this would be a monomer of RNA. How do I know? Because there's an OH right here. What about this one? This would be a monomer of a polysaccharide. It would have a glycosidic bond. This would have a phosphodiester bond, I should say. What kind of bond would hold them together? This one over here, this is protein. This is an amino acid, so it's part of a protein. It would stick to its neighbor with a peptide bond. Number 5, draw the interaction of water molecules with a sodium ion. That would be a hydration shell where the negative water is facing towards the positive sodium. Number 6, name the functions of membrane proteins. That's just a list. You can look that up, but that'll be on exam 2 for us. Number 7, a patient is suffering from a buildup of old organelles. Which organelle is malfunctioning? Well, which organelle digests old lysosomes? Oh, I screwed that up. Which organelle digests old organelles? Lysosomes do. Okay, lyso, that means to break down, right? Lysosomes. Draw a carboxyl group. That's a carbon double bonded to an oxygen on one, and then also with a single bond to another oxygen. That's a carboxyl group. All right, so that's an example of exam 1. Your exam will be about 50 questions long, and we'll have questions about 10 to 15 questions from each chapter. From starting at, starting at the chapter 1 and ending with the chapter on organelles.